Now I could like to begin with our 29th session and entitled SCIENTIA today, which is entitled as MIcrosponge Drug Delivery System. And it gives enough pleasure to introduce our speaker to today's session, Amol Gholap sir. Sir is currently working as an assistant professor in Department of Pharmaceutics in St. John's Institute of the Pharmacy and Research, Palghar. He has completed his B Pharm and M Pharm from Savitribai Phule University and currently pursuing his PhD in pharmaceutical science from Pune University. He has published some research and review papers in national and international journals. He has received 2 grants for minor research projects from Mumbai University and currently guiding 2 MPharm students, quality assurance students. His key area of research includes narrow carriers lipid-based drug delivery system, including nanosponge, nanogel, nanoemulsions, etc. With this brief introduction, I now request Sir to take over the session.
Thank you so much for such. Thank you, Samiksha, for such a nice introduction. I will share my screen. Okay. So today I'm here to present a seminar on MicroSponge drug delivery system. These are the content of my presentation, which includes advantages, drug release mechanisms, one case study, etc. So these are the content of my presentation. So first of all, what do you mean by MicroSponge? MicroSponge is generally a novel drug delivery system which is used for the topical drug penetration through the skin. Normally, these are the spherical cross-linked polymeric system having some pores and sizes range from 10 to 25 micrometer. We know that most of the drug when we apply topically on the human skin, they are having a problem of skin penetration because of the stratum corneum issues. So when we compare this MicroSponge with the contemporary delivery systems like Liposomes and microcapsule, it is also having the ability to give the drug through the skin because of their size. Size of the human skin is human skin pore are from 500 micrometer to less lower to the 250 micrometer, and this MicroSponge is having a size of 10 to 25 micrometer, so they can easily pass through the human skin pores and they can release their content over there. Now, why we need to use the MicroSponge? Because of the some of the problems which are arise in the conventional topical drug delivery system. Conventionally, we can use ointment, gels, creams for the topical system, for the topical treatment of the many type of diseases. These are suffering from 2 basic problems. One is the having a problem of over medication, and second is the under medication. Here, when the drug is overloaded on the skin, then it takes some time to get within the skin pores. If the skin penetration is less, there is an issue of under medication. So the drug which is present inside the skin is not able to achieve its concentration into the therapeutic window, and there is no any good effects from the drug. Second is the problem of over medication. In the over medication, there is a burst release of the drug from this type of contemporary delivery system, and because of the burst release of the drug, because of the overcrowding of the drug at the local site, there is a problem of skin irritation. Now, the dermatological layers are extensified with the different types of sublayer systems, and different type of topical drug delivery systems are available along with the MicroSponge. So we'll see one by one what are the systems are and what are the drawbacks of that system, so that we require to have this MicroSponge. Now, currently, we know that the polymer micelles, microemulsions, and liposomes, these are lipid-based drug delivery system. Now, there is a combination of lipid and colloidal drug delivery system for the topical delivery of majority of the drugs. First of all, when we use lipids as a drug delivery system, this lipid generally improves the adhesion of the drug. Second, the lipid which is we are using over there, this lipid can get exchanged with the lipid from the skin, and there is a replenishment of the lipids within the skin. So exchange of the lipid content is one of the major factor which will help us for the drug delivery system. And the bioadhesion which is given by the lipids is also helping for the delivery system. Third important point is that when we use the lipid, we can able to transfer this lipid into the nano size or micro size, so we can fluctuate the particle size depending upon our need. So there is a scope to increase or decrease the surface area depending upon the change in the particle size, and that is also easily possible through the lipid-based drug delivery system. The majority of this lipid are actually increase the skin hydration. So when you increase the skin hydration, there is a increase in the skin penetration of the drug. So these are the example of some systems which are having some of these effects. First one is the polymer micelles, second one is the microemulsion, and third one is the liposomes. Now, polymer micelles are actually look like this. So these polymer micelles are used to target the different types of oil. Actually, these are the oil-based carrier system. But the polymer micelles are suffering one of the major issues, that when the drug is get entrapped within the polymer micelles oil, then there is a problem of aggregation of the oil droplets. Now, second issue is that when the aggregation of oil droplets is started, then the drug which is present over there, it loses stability. So the aggregates are going to be increase their size because of the more number of aggregation that reaction is started, and the stability issue will get arises. Second issue is that if you have a solubility issue of some of the drug into the oil, we can use the surfactant as a solubility enhancer. But surfactant themselves are using in a high concentration, and because of that, some papers are reported that this high concentration of surfactant produces some of the toxicity issues. So there is a payload, we can say that disadvantages to the polymer micelles. We cannot able to load the high quantity of the drug through these micelles. And second, there is always a problem of stability of the drug within the oil droplets. Second are the liposomes. Now liposomes, now we have a surface modified liposomes. But initially when the liposomes were there, without any modification, at that time they were highly acceptable because of their biocompatibility and having a low toxicity. But after all, many researchers have reported some of the major drawbacks of these liposomes. That's why there is a need to have a surface modification or change in the trigger system of the drug release from the liposomes. One of the major disadvantages of the liposomes that there is a storage condition of liposomes is there. We require a certain temperature, 2 to 8 or 2 to 10 degrees centigrade to store the liposomes. Liposomes temperature when get increased after 45 degrees centigrade, then it produces the leakage of the drug content from it. And because of the leakage of the drug content, there is no any control release fashion is available to monitor the drug. Second issue of the liposome is that these liposomes are actually having a scale-up issues. Means on laboratory, we can prepare the liposomes with a good number of results. But when you scale up for the industrial application, in that case, there is a problem of stability of the drug within the liposomes because of high viscosity. And the third important thing is that we cannot able to load more amount of the drug because of the lesser amount of intrahepatic efficiency of the drug within the liposomes. So these are the liposomes where we can have the phospholipid bilayer and internally we can entrap our drug. So because of this issue, the liposomes are not one of the favorite drug delivery systems for the topical route. Then after that, we have the microsponge. Now, what are the advantages that are present for the microsponge? It absorbs the oil 6 times its weight. So as like we have a routine sponge which we are using in a household, this sponge is having the same type of property of oil absorption. So if you want to have a drug which is soluble into the oil, that oil can able to get entrapped within the microsponge easily because of this property. It shows over a 12 hours of drug release. So sustained release drug release system can be achievable. Then physically, thermally, and chemically it is stable. It is non-irritating, non-mutagenic, non-allergenic, and non-toxic to the skin. Most of the polymer which are used in a microsponge preparation are biocompatible. For example, we have a ethyl cellulose, then Udragit RS100, RL100. All these polymer cyclodextrin also there. So all these polymers are biocompatible. So there is no any issue of irritation, mutagenicity, or allergenic condition towards them. And majority of the content of the microsponge is actually filled by using the fillers which are biocompatible. So there is no any case of toxicity also. Then majority, one of the major application of the microsponge is that we can convert this sponge that is liquid into the powder. And once we have a lyophilized powder with us, we can able to form n number of dosage form for the topical delivery system. Means we can prepare a microsponge entrap gel, cream, ointment, a hydrogel, depending upon our need. So the flexibility for the selection of dosage form is also given by the microsponge drug delivery system. Now the next point is method of preparation of microsponge. Now there are the two methods which are used for the preparation of microsponge. Okay. First method is called as liquid-liquid suspension polymerization, and this is the one of the prototype diagram which is used for this preparation. And second one is a quasi-immersion solvent diffusion. Now in a liquid-liquid suspension polymerization, we require to add the monomers, then we require to add the stabilizer, drug, so along with the cross-linkers. This reaction of the polymerization, that is where the monomers are attached with each other, and the cross-linking is done by the cross-linker, is achieved by using addition of some catalyst. Change in the temperature also act as a reaction initiator for this case. But one of the major problem of the liquid-liquid suspension polymerization is that here there is no any guarantee that 100% added polymers will form the polymers. If the 100% monomers are not transferred into polymers, then after the end of this reaction, we will have some monomer residues. So these monomer residue, which are not transferred themselves into polymer, are act as a or get entrapped within the polymer chain. So it is very difficult to actually figure out which monomers residue are intact and which monomer residues are abstract, which having a no use into the microsponge drug delivery system. So these monomer residue are actually creating a problem of drug release from this system. So we require such a system where there should not be any residue problem. And for that reason, the second type of method is used, that is called as liquid-liquid suspension polymerization or quasi-immersion solvent diffusion. Now in quasi-immersion solvent diffusion, the majority of the polymers which are used are ethyl cellulose and Udragit. Now here, actually, these methods suggested that microsponge is the group or bunch of microsphere. Initially, they used to prepare a microspheres. These microspheres are combined with each other and they will form the microsponge. Now, initially, when you heat something within this, with the help of the temperature and some core forming agent like dichloromethane, then ethyl cellulose like polymer, they form a balloon. And when they form a balloon, they get entrapped the drug and dichloromethane within their shells. So whatever the dichloromethane which is entrapped within the shells, it coming out of the shell by making a pores to it. So the pouring through the pouring agent is happen because of the high speed rotation or high speed homogenizers which is we have provided, and the compatibility of this size is actually monitored through the pressure. Now this is one of the more promising method for the preparation of the microsponge, because when we evaluate the particle size of the microsponge, then there is a uniform mixture will get. Second is that the entrapment efficiency also get improved for this method, and there is no any residue of monomer issue which was there in earlier case. So this is one of the, we can say that diagrammatic presentation of it. Now, how the drug is going to be released from the microsponge. Now, there are the two ways by which the drug is released from the microsponge. First one is a temperature change, and second one is the pressure. We know that when microsponge is the porous in nature, so majority of the drug goes within that pores, and it get inserted within the microsponge. So such microsponge when get entrapped within the cream or gel, and when the customer or patient is applying that gel on the topically with the help of some pressure, because of the pressure trigger squeezing, the microsponge gets squeezed and it will open up, or it will give the drug from its pores. So it is said that it is one of the way to actually take out the drug from the microsponge from inside to the outside. But majority of the studies shows that in the prior means 1 to 2 squeezing only gives 50% output of the drug from the actual drug loaded. So second way of the drug release is temperature change. Now, if you apply the change in temperature, there is a change in the release of the drug from the microsponge. Release study, if you assess through the kinetic models, then we have a different kinetic model. For example, we have a first order release, second order release, zero order release, then Higuchi model, Polymer-Micelles, like this. So every model we are going to evaluate it by using the two types of parameters. First parameter is a regression value, regression coefficient, and second value we have that depend upon the diffusion coefficient. That is also called as diffusion value, that is n. So this diffusion value n is having a range from 0.5 to 1 and 1.5 to 2. Depending upon that value, we can assess whether the drug is released through the first order, zero order, Polymer-Micelles or Higuchi model. And most of the papers which are there, which we have done the research on the microsponge, they have shown that drug release mechanism from the microsponge is generally follows Higuchi models. So Higuchi is a controlled drug release models which suggested that there is a non-Fickian system and Fickian system which are available over there. So the diffusion is one of the major role which helps drug to get released. So there is a controlled drug release fashion is available from inside to outside and from outside to the skin. Now, some of the literature survey shows these are the one of the major of the topical active drugs which are formulated into the microsponge by using these polymers, and these are the solvents which are used for them. We can see most of them are antifungal drugs, and the solvent which are used are generally dichloromethane. Few papers which are present over there, they have shown that the use of the dichloromethane should not be prompted because dichloromethane is toxic to the human skin. And when we use dichloromethane as a pouring agent, in that case, we need to check whether the dichloromethane is completely get exhausted from the microsponge or not. So for that reason, we require to calculate residual solvent content. And some of the papers have shown that when we calculate the residual solvent content for the dichloromethane, some traces of the dichloromethane are still there. So in that case, we require to increase the stirring speed or high pressure organizer parameters we require to check so that we can able to exhaust the total DCM from the system and will decrease the probable toxicity that may arise because of this content. Now, these are some of the marketed formulations of the microsponge. These are some of the manufacturers. Currently, these dermic laboratories which are there, which have the Carac cream, dermic laboratories are having a mini products of the microsponge, and these are having a n number of applications for the topical drug delivery systems. Then we'll discuss one of the case study so that we will actually figure out how the drug is going to be monitored through the microsponge, how the microsponge were prepared, and which type of systems are used for the evaluation of the microsponge. So the case study that was published into the therapeutic delivery journal, London. the authors, they have selected the drug ibuprofen nitrate. Now the ibuprofen nitrate, this is actually, they have reported it is around 1,000 times more effective than ketoconazole, which is one of the standard drugs used for the antifungal regime. But the ibuprofen is having an issue of oral bioavailability. So in the rationale, they have mentioned that this antifungal drug, that is ibuprofen nitrate, is having a less oral bioavailability. So only 30% is bioavailable. And second issue is that if you prepare a normal ibuprofen nitrate cream, then it having a issue of less percutaneous absorption to the stratum corneum. So one of the group of scientists, they have prepared microcapsule of ibuprofen nitrate, and they have reported that there is a formation of, there is a more irritation is arise because of the burst release of the ibuprofen. So there is a need to have effective topical delivery of the ibuprofen nitrate because of its oral bioavailability issues. Second, there is a need to address the irritation issue that is there because of the burst release of the drug. And third, there is a need to increase the skin penetration for the effective percutaneous absorption. So for these three reasons, the group of scientists, the group of authors, they have prepared or they have selected MIcrosponge drug delivery system as a topical delivery for the ibuprofen nitrate. Now, they have selected some of the factors. Quasi-immersion solvent diffusion method they have selected. Then these are the objectives they have planned. In that, they have mentioned that the stirring speed, stirring time, amount of the polymers, then volume of this internal phase, these four factors they have selected for the effect purpose. Now the polymer they have selected was ethyl cellulose. The pouring agent or solvent for the drug they have selected was dichloromethane. Stabilizer was polyvinyl alcohol, and water is used for the solubilization of PV. Now, for the processing factor, they have selected stirring issues, and for the formulation factor, they have selected amount of the polymers. So we can say that they have given an equal stress for the formulation factor also. Two of them they have selected and two for the formulation factor. And they have planned to have the skin permeation study, antifungal study, skin irritation, skin penetration models by using the rat skin. Now, these are the plan of work they have done. Now, first of all, they have given this total drug profile of this ibuprofen nitrate. Preliminary screening they have done by using the ultraviolet spectroscopy, by using physical parameters like melting point, IR spectrometry, then differential scanning calorimetry, and PXRD. Now, these are the PXRD and DSC. These are the two tests they have done to actually check what is the behavior of the drug within the microsponge and without the microsponge. Now, DSC is the test which we can use to actually check is there any thermal change of the drug when we interact it within the microsponge. Without microsponge, we can say the drug is having its own peak at melting point by using the DSC technique. And when we prepare the microsponge entrapped drug, then again we require to take the peak of the same drug into the dosage form, into the formulation. And within the formulation, if the peak is get shifted or there is a change in the peak value, means suppose it was on 133 degrees centigrade and now it becomes 180 or something like that, it indicates that the drug is not stable anymore within the sponge. Second is the determination of physical state by using the PXRD, where we require to have a change into the peaks of this drug so that we can confirm these peaks are going to be changed because of the encapsulation of the drug within the ethyl cellulose. Now, preliminary study they have done like this. These are the 17 batches they have prepared. Each batch they have coded from M1 to the M17. This is ethyl cellulose concentration, PVA concentration, and rest of the parameters they have added. Then they have done the study of drug polymer ratio. What is the impact of that ratio? They have changed, and these are the limited number of the batches they have selected, which having a good number of physical properties. So the preparation of it, after the MicroSponge preparation, they have prepared the gel by using 1% carbochor 934 and 1% triethylamine. The prepared gel they have neutralized at pH 7, and these are the characterization of this MicroSponge they have done. Now we'll see only a few characterization steps. So here they have done the physical characterization of this ibuproconazole nitrate to actually calculate the absorption maxima of it. These are the 237.5 nanometer. That was the absorption maxima they have reported, and they have selected these two types of solvent also. These are the graphs they have given. Calibration curve, to draw the calibration curve, they have selected this range. In distilled water and phosphate buffer pH 5.4, phosphate buffer solution pH 5.4. Saturation solubility also they have actually calculated. So these are the IR spectrum values. So from this IR spectrum, I have put it only few number of graphs over there. So they have interpreted that when the system is there within the MicroSponge, there is no any physical challenges to the drug. Physically, the drug was stable into the formulation, and it was actually proven by the DSC and IR. So these are the graphs of the PVA, and this is the compatibility study. Over here, we can see there is no any big change into that. So these are the microscopical images of the slides they have put it over there, and this shows that there is a n number of changes into the factors, and few factors which they have selected. When they change the factor, their ratios, these 6 different types of formulation they have obtained. So in the first case, we can see there is some of the drugs they have said that the drug is entrapped within the MicroSponge, which is spherical, but few of the drug MicroSponge they were changed. So there is a fabrication issue is there. Second, there are some of the droplets they have mentioned. So this is the issue. In third, the sphericals are good, but these are not having a proper type of size. In D, that is 4, they have said that majority of the MicroSponge get ruptured. 5th and 6th, they have mentioned were the better batches, and they have carried forward this project by using the E and F batch results. So in the particle size analysis, particle size distribution, this graph they have added over there. Now in particle size analysis, if you require a monoporous and polyporous, then we require to have the value of this particle size distribution. If it is within the range of less than 0.5, we can say that more number of polymers are having uniform size. These are monoporous. But when the size is extend from 0.5 to 1 and more than 1, here we can say that there is a polyporous. Means some of the MicroSponge having a bigger size, some of the MicroSponge having a lesser size. So this is the scanning electron microscope images they have given. So here we can see these are the spherical MicroSponge they have obtained. And when you zoom it, then actually we got this type of MicroSponge, that is single entity of this MicroSponge, where we can see the pores are there. It is porous. There is a space available within the MicroSponge where we can see the drug can able to add it over there. And these images shown that whatever the pores which are present over there, most of the pores is having the same size. So this is the PXRD. Here we can see some of the crystals get changed over here. It indicates that the drug is encapsulated. Because of that, because of the encapsulation by the ethyl cellulose, the drug is not able to give its spikes. These are the entrapment efficiency values and the gels. So here they have mentioned that the entrapment efficiency values is actually more into the M3 batch, and the gel is also having a more entrapment efficiency values. So these values they have reported having a good number of more percentage of entrapment efficiency as compared to the other type of drug delivery carriers. Then this is the in vitro drug leads, and through the in vitro drug leads they have mentioned the Higuchi. Their studies follows the Higuchi models, and likewise I have said that most of the studies when they reported the regression values, the Higuchi is one of the more we can say that kinetic release drug release models that was applicable over there. So it indicates that there is a control release of the drug from the skin. These are the diffusion study they have mentioned by using the in vitro diffusion method, by using dialysis membrane, and by using the ex vivo drug diffusion by using the rat screen. Here we can see they have compared with the marketed. So in marketed, the drug release high percentage is only 50%, where in the batches of M1, M2, and M3, we can say the highest is M3, where the drug release is around 95%. Same in the case of ex vivo drug diffusion determination, where the marketed is gets stuck near about 40%, where the drug release from they have selected batches, they have shown it is crosses more than 60% of the drug release. So there is a substantial more impactful release of the drug from their selected formulation as compared to the marketed cream. So this is the in vitro antifungal activity they have done by using a zone of inhibition study. This is the reported zone of inhibition in centimeter they have reported. So here we can see the batches which they have selected, they are having these zone values, and 1.23 centimeter is the highest value of the zone which is reported for this batch. In ex vivo skin penetration study they have done, and these are the values they have given for the 6 hour. For the marketed, the values is obviously lesser, that is 2.29 as compared to the 3.10 for their batches. And this is the major skin in vitro skin irritation study. So here they have reported that one of the major pillar for the rationale is to inhibit the skin irritation of the drug of ibuprofen that was induced because of the burst release from the microcapsule. So here they have selected the animals, that is rat skin. On the live animals, they have done this study. So on the rat skin, after deshaving it, 4 centimeter square area were selected, and on that area they have applied the control, no any drug or marketed formulation they have applied, but on the selected animal skin they have applied the formulation gel, and one of the rat skin they have applied with the marketed gel, and they have checked is there any skin irritation issues are there or not, and they have done the inspection of these for totally 7 days. So they have found that there is no any skin irritation done after the 7 days. But in marketed one, they have found that there is a little bit slight irritation was there through the red patches that was observed on the day 6th and 7th day. So summary and conclusion, they have reported that the rationale that was they have selected for the beginning of the study that was achieved. So there is no any skin irritation issue that was there by using the MIcrosponge drug delivery system. Second, they have selected some factors from formulation and from processing backgrounds. So they have said that the concentration of the drug, then the polymer concentration and DCM concentration, these three are one of the major factor for it. From the from the processing factor, the stirring speed is one of the major factors. So they have done the comparison for all the study by using the different tests like SCM, DSC, and all that. And lastly, they concluded that whatever the batches they have selected, that M1, M2, and M3, that able to give or deliver the drug release for at least 12 hours with more than 60% of the drug release as compared to the marketed formulation, and there is no any irritation issues. So it proves that the drug delivery system is actually applicable with minimum number of side effects when it is delivered through the MIcrosponge drug delivery system. And these are some of the reference I have used for my presentation. Okay, thank you. If you have any questions, you can ask.
You can drop your questions in comment box.
Okay, thank you all.
Good afternoon, everyone. I feel immense pleasure to take this opportunity to propose a vote of thanks to our eminent speaker, Amol Gholap sir, who graced this webinar with extremely relevant address on MicroSponge drug delivery system. Sir have rightly highlighted on various points like what are MicroSponge, need for MicroSponge, method and preparation of MicroSponge, how drug release from MicroSponge, and also discuss one case study and how the MicroSponge are evaluated. My deep sense of appreciation to the sir and thanks to all the participants who attended this webinar with a great willingness. Thank you all. We will be starting another session.